So in our preclinical mouse studies, we used a mouse that has a mutation sort of in the middle of the dystrophin encoding gene. And the presence of that mutation prevents the mouse from being able to make a full-length protein. It has the same loss of expression of the dystrophin protein, although its disease is more mild than humans with Duchenne muscular dystrophy. It still has loss of muscle function. We used CRISPR-Cas to make 2 cuts in the DNA, flanking on either side of that mutation. That resulted in the removal of that section of the gene, and now the gene could be read in a way that would create a protein that lacked that sort of middle internal part, but still could go from end to end and have partial function. And we were able to show at a molecular level that we could rescue gene function, both by expression of the protein, to the presence of the protein in the muscle, and also by an improvement in the force that the muscles in those treated animals, those treated muscles, could actually generate.